Hi everyone. So now that we've talked about what sugars are chemically and how they are extracted, um, manufactured, etc., now we're going to talk about uh, the functions of sugar, how we actually use sugars in food, and then we'll talk a little bit about nutrition. So the first function of sugar in food preparation is the most obvious one. Is that sugars are sweeteners. Um, and in general, the more simple a sugar is, the sweeter it is. Um, so we haven't talked about complex carbohydrates, but complex carbohydrates are things like starch, and you've probably heard of starch before and know that it's not sweet, right? Like think about a potato. Potato is not sweet, and that is a more complex carbohydrate than all the simple sugars that we've talked about. I have already mentioned that fructose is sweeter than any of the other monosaccharides. So this table here is listing several different types of sugars as well as sugar alcohols and comparing their relative sweetness versus sucrose. So sucrose gets a 1 because compared to sucrose, it's a 1-to-1 ratio of sweetness. And fructose is nearly twice as sweet as sucrose. Um, and um, I do want to point out maltose, I said in the last lecture, was the least sweet. That's what the book says. And then the book has a table that contradicts itself. So lactose is less sweet than maltose. So there's that correction there. Maltose is not the least sweet um, disaccharide. Or least sweet sugar. Um, so yeah, glucose is almost as sweet as sucrose, but the big thing to know is fructose is quite a bit more sweet than sucrose. And what determines sweetness is something that like scientists have called the triangle of sweetness. It's not a literal triangle, and the book almost makes it sound like it is. But essentially, in the taste buds, this is the taste receptor, and there are 3 binding spots, spots on the receptor that want to bind to a sugar molecule. And the closer though that sugar molecule hits having certain spots on the molecule that fit nicely and bind with those specific spots, the sweeter it will taste. Okay. So it might not fit quite as, if it doesn't fit quite as perfectly, then the sweetness is less. If it doesn't fit at all, you wouldn't taste any sweetness. The second function that sugars are used for in food preparation is preservatives. Let me look at, yeah. Um, so, and this comes from the fact that water is drawn to sugar molecules, right? So water is polar, and sugar molecules also have polar parts, which is the hydroxyl groups. And because this water is drawn to the sugar molecules, um, single-celled organisms cannot access the water, as long as you don't have more water than what can be drawn to the sugar molecules. Okay, so this is why something heavily sugared, like fruit preserves, jams, jellies, syrups, etc., even though there's actually quite a bit of moisture, quite a bit of water there, the water is all being drawn towards the sugar. Um, and what happens is if bacteria are present, um, those bacteria have a lot of water in them, all right? And their cell walls are very permeable. Water can go in and out. Well, if the water is being drawn towards the sugar. Um, this is called osmosis here. So like this here you can think of as a like a kind of a cell wall. And the um, the water is going to be drawn toward that concentrated solution of sugar and out of the non-concentrated solution, I should say, with quotes, inside the cell. Okay, because it wants the it wants sorry, it wants equilibrium to happen for essentially the dilution of the water to be the same in both places, right? And here you can see this kind of like the cell. This is very dilute. Well, here it's not. And so this stuff is not going to go into the cell because it's too big. It won't just go into the cell wall, but the water can freely go out. And so what happens is you get a desiccation, a dehydration of the bacterial cells, and they can't grow at minimum. And ideally, they even die. So that is how it preserves, sugar preserves some foods. Sugar also helps baked goods stay moist for the same reason. So it's related to the same property where sugar is holding on to that water. So it keeps the water from evaporating as easily. It's holding on to it more tightly so it can't be lost to evaporation in the air. And this property is better if you use invert sugar versus sucrose, but sucrose is still pretty good. So one of the things, it's like the cat is going to make an appearance in every video, y'all. So one of the things that you can do to prevent cookies from going hard, or if they have already gotten hard, you can actually soften them, is by putting something in your container of cookies that has less sugar than the cookies, or brownies or cake or whatever. Although with the cake, you might end up making it soggy. I'd hesitate on the cake. But you add something that has less sugar in your container, such as the slice of bread, and what's going to happen is the higher sugar content in the cookies is going to steal the moisture from the bread. And that will keep your cookies staying moist longer, or if they have already gotten hard, they can go back to being moist by stealing the water from that bread. The third function of sugars is as a tenderizer. And we're not talking about meat, we're talking about baked goods here. So in baking, you are balancing the ingredients that give structure, such as flour and eggs, with ingredients that weaken structure. And those would include sugar and fat. Okay, so flour, eggs give more structure, sugar, fat weaken structure. And adding more sugar to a recipe results in the proteins in the flour and the eggs forming weaker bonds. And this creates a more tender, softer crumb to a cake. The batter will pour more easily and you'll have a greater rise. And so essentially more sugar interferes with the flour's ability to form an elastic structure. Now, however, if you add too much sugar, the structure can weaken so much that you have collapse. The gas that is created, during your leavening process will just be able to escape, and you actually can end up with a dense cake, opposite of what you are trying. So you can see this here in this little experiment that I found that somebody did online. So they have this pound cake recipe and they tried using 75% of the recipe, 100%, 125%, 150%. So just increasing the sugar only in the recipe. And so you can see that this 125% sugar formula had the best rise. And they said it actually was the best tasting too, and it was the most tender, most moist cake of the four. But once they went over a certain point, and so they did 150% of the sugar called for in the recipe, they lost all of that benefit, and it collapsed enough that you ended up with a dense cake that, I mean, actually is pretty similar to the 6-ounce and 8-ounce sugar, but of course it's going to be sweeter. I think they said that this one also ended up really sticky because at some point, by this point here, they had more sugar than what could be bound to all of the wet ingredients, and so it precipitated out and ended up with like a sticky outside of the cake. Okay, so now fourth function is a crystallizing agent, and we're going to spend a lot of time on this one. So fourth one is not the last one, but we are going to spend quite a bit of time on it, so much so that when I get back to functions, you're going to kind of have to be like, okay, where am I again? Just a warning, crystallizing agents is a long section, but it's still in a list of functions. So sugar dissolves in water because of the large number of hydroxyl groups. Okay, these are all the polar parts of the sugar molecule. Okay, and we've seen these hydroxyl groups last week. So, and actually what I'm circling here is not the hydroxyl groups, it's the water. Okay, so the hydroxyl groups, OH, 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 right? You see them all over the sugar molecules. And so this right here is a sucrose molecule. And what you're seeing here is that the oxygen and hydrogen in the hydroxyl group are a polar covalent bond. Just like oxygen and hydrogen form this type of bond in water, H2O. So the oxygens in the hydroxyl group have a partial negative charge, and the hydrogens in the hydroxyl group have a positive, partial positive charge. And so they are going to bind with the water via hydrogen bonds, and so the oxygen is going to bind with hydrogen, and the hydrogen on the hydroxyl group is going to bind with oxygen. Okay, and so you see that all over. Sugar loves water. Okay. So, and that is why it dissolves in water. But only so much of a solute, or sugar, can be added before a solution is saturated, before it has bound to all the water it's going to be able to bind to, and any extra sugar that you add is just going to sink to the bottom because there's no water left for it to bind to. However, if we heat that solution, that increases its ability to dissolve. It's able to bond to more molecules before it becomes saturated. Okay. And so it's really just kind of a property of how fast things are moving and how it's able to just grab on, like what are the odds of it interacting with the water molecules when they are cool versus heated, right? There's more moving around, more bumping into each other. There's more likely for a bond to be able to form. So, if the solution then cools down, though, that solution is what we call supersaturated. I've changed the definition on you for this one, so it's red. And so what supersaturated means is that we now have a homogeneous mixture where the concentration of the solute, the sugar in this case, is greater than the maximum amount that can dissolve at a given temperature. Okay, so we've cooled it down and it doesn't all just come out of solution and sink to the bottom. more of it is still remaining dissolved. Now, it's not going to be as stable. And different um, molecules, when they're supersaturated, are more or less stable than others. So, for example, salt. I can do the same kind of thing with salt and dissolve more salt in warm water versus cool water, but salt is not going to be is not stable at cooler temperatures when it's super saturated, and so salt is pretty much immediately going to precipitate out and just sink down to the bottom if I cool it down. But sugar um, is better at remaining supersaturated. It's a little bit more stable. So if you start out with a certain amount of sugar that is soluble in the amount of water you have, okay? And then as you heat it, that solubility is going to increase. But also the water is evaporating, okay? And so I haven't changed the total amount of sugar that is in my solution, but the concentration of the sugar is increasing because I'm losing water, okay? So the heat continues to rise and the solubility continues to increase, its ability to dissolve, and water is continuing to evaporate. And this is creating a supersaturated solution as we do that, okay? Because the concentration is increasing because we're losing water. And it's able to do that because the heat keeps rising, okay? So this is kind of the principle of heating up sugar, especially for candy making. Okay, so some candy, and this is a little note different from the book. I think the book might even say all candy, but not all candy. Some candy is made when sugar crystals separate from a supersaturated sugar solution during cooking, or during cooling, I mean. So rock candy, which is what we're basically making this week in lab, is made by slowly allowing large crystals to form in a supersaturated solution. So we are putting in this bamboo stick that has been seeded with sugar, and it's in a supersaturated solution. And I told you that supersaturated solution is not super stable. And so as soon as we add seeds to that solution, it's going to start forming crystals. And it does it pretty slowly with rock candy. Now, fudge, on the other hand, we use stirring to increase the number of crystals and therefore make the crystals smaller. And so our goal in fudge is a very small crystal, but we are having those crystals separate from the sugar solution, just like rock candy. Well, not just like it, but now hard candy. Hard candy, there's no crystals. We have the supersaturated solution and we cool it down so quickly that no crystals can form. Cotton candy is also superheated sugar. The liquid is sprayed out and it cools so quickly that no sugar crystals should be able to form. So not all candy is made from crystallization. But understanding crystallization is important to making all candy. Okay, so I already mentioned seeds. Let's describe that a little bit better. Sugar crystals like to form around particles that enter a sugar solution, and those particles are called seeds. A seed in sugar terminology is really anything that the sugar molecules can attach themselves to form crystals. It can be a sugar crystal that is placed in there. So for example, like we talked about last week, with the sugar that is added to the sugar solution in the making of sugarcane sugar, sugar from sugarcane. It can be things that you don't necessarily want to add, so it can be lint or dust. It can be a rough surface. It can even be an air bubble, can cause some of the this crystallization. And air bubble is the seed. So controlling this crystal formation and the size of the crystals that we form is the goal of all candy making. Okay, so not all candy making are we wanting to make crystals, but all candy we need to control that. All right, whether that is because we do want to make big crystals or tiny crystals or lots of crystals or very few crystals or no crystals at all. That is our goal in candy making, is to understand that and control the crystallization. So the factors that affect the crystal formation, okay, so this is kind of a separate list within the crystal formation category of sugar functions. The type of sugar. Remember that invert sugar, so sucrose is most commonly used, but invert sugar results in slower crystallization. So remember that invert sugar is just the two monosaccharides that make up sucrose, which are glucose and fructose, and a 50-50 mixture of those. Okay. And the reason invert sugar results in slower crystallization is because monosaccharides are more soluble in water and they form smaller crystals. Okay. And invert sugar can be produced by adding sucrose and then adding acid to sucrose and then neutralizing the solution. Okay. Second factor that can affect crystal formation is the use of interfering agents. Okay. These are any substance that can prevent or slow down crystal growth. And so those can include corn syrup, okay? So corn syrup is high in glucose. And so this basically is the same principle as the type of sugar, right? It's smaller crystals because it is a monosaccharide predominantly. Almost completely, because corn syrup is cornstarch that's been converted to glucose. Okay. Then other interfering agents are any ingredient that is high fat. And so what's happening there when we add butter or cream, those fat molecules are suspended in that solution and they're keeping those sugar crystals apart. They're keeping them separate. So it's just really like a physical barrier preventing those sugar crystals from meeting or the sugar that wants to bond to the sugar crystal from meeting and joining up to add to the sugar crystals. And so high fat ingredients will lead to a smoother, creamier candy. And third would be high protein ingredients. So an example there would be egg whites, and similar function to the fat. It's really just a physical barrier. Notice the words are pretty much exactly the same, except I say protein instead of fat. They suspend and separate the sugar crystals. So again, we end up with a smoother candy. Think about macarons. Okay, and then finally acids. So cream of tartar, for example, or vinegar. This is actually kind of back to this same principle of type of sugar, because what the acids do is hydrolyze the sucrose, resulting in invert sugar. And so then we have the monosaccharides and less tendency to form crystals, and the crystals are smaller. And these are not necessarily a bad thing, right? We've specifically used some of these interfering agents when we don't want big crystals, or if we are trying to prevent crystal growth to begin with, okay? The third factor that can affect crystal formation is agitation. And so agitation would be the beating and stirring of a candy solution, and I am adding here to that definition, or any mixture. Okay, I mean that's kind of obvious, right? But the phrase at the term agitation does not specifically refer to candy. Um, so principle here is okay. So you have a hot solution, and to start with, right? Because you are supersaturating that sugar. And any process is going to happen faster when hot. So what happens with agitation is that stirring, whether hot or cold, promotes production of sugar nuclei or seeds. Okay, it promotes the production of those first seeds that start the crystal forming. Uh, and so this crystal formation is going to occur faster at warmer temperatures, just like any chemical reaction occurs faster at warmer temperatures. Um, it also will occur faster if the viscosity of the solution, how thick it is low. Okay, so a hot sugar solution, it has two things going against it if you aren't wanting crystal formation, or two things going for it if you are wanting this crystal formation, is that it is warm, and so chemical reactions are faster, and it is not viscous. Okay, so there's nothing slowing down physically how fast the molecules in that solution are moving. Now, as it cools, the viscosity is going to decrease. Um, and it's going to be harder for those sugar molecules to move around, just physically harder. And that therefore is going to slow the reaction down even more than just the temperature change alone. So in a cool solution, we create more sugar nuclei through agitation. Um, and So like if we're waiting, if and what I mean by that is we're waiting until it's cooled to stir, we are going to create more sugar nuclei through that sugar seeds through that agitation. Um, and the reason we'll do that is that it controls the crystallization process. There are more nuclei, but because it's cooled and because it is more viscous, more thick, um, there only so much sugar is actually going to be able to crystallize. And so we create more crystals, but we're preventing them from becoming too large because you keep stirring until that temperature and the viscosity are at a point where where new crystallization is no longer happening. And so what you end up having, and what we're really talking about here, like this is going to sound very familiar, that what we did with the fudge. And so what we're doing is we're creating lots of small crystals. If you don't stir the fudge at all, you're, well, let's go back. So this is why we had the specific steps in our fudge making lab. While the solution is heating, and then after we took it off, we did not stir. We did not want to start forming crystals yet. Stirring would have created sugar crystals prematurely. That would have gotten out of control because everything is too hot and can react too quickly, and it would have led to grainy fudge. You would have felt those sugar crystals in your mouth when you take a bite of them. And hopefully we feel no sugar crystals in our mouth when we try it this next week. But so what we did instead is we waited until the temperature was low enough that we could control that process and create lots of small sugar crystals. And so what would happen if we didn't stir at all? What if, like, we don't want any sugar crystals? So it wouldn't crystallize at all if you didn't stir, and you would have liquid fudge. So the sugar crystals actually provide some solidification to that fudge too. Okay, fourth factor is cooling. Okay. Um, so, and the key thing, one of the key things here is not doing that stirring that we talked about with agitation, not doing that too early or late is important. And cooling quickly can prevent crystals from forming. I think the book mentions something about taffy candy, and it makes it sound like it is a crystalline candy. I find it. Yeah, fondas and taffies are often cooled on a marble slab. Well, taffy is actually not a crystalline candy. You aren't trying to form sugar crystals in taffy. I don't think you've ever felt a crunch of sugar when you've eaten saltwater taffy. Um, and actually, invert sugar or corn syrup is used to make it. So they're specifically not trying to form sugar crystals there. Um, and so there's not really a big danger of forming big sugar crystals because of the use of invert sugar or corn syrup in making it. But taffy or fondant on a marble slab does cool it quickly. Now, fondant is made from sucrose and does use the marble to cool it quickly, but you also are constantly working it while it's cooling. So they'll like have it on the marble and they'll be using their little spatula and they're just constantly turning it and working it. And so there is crystal formation, but kind of like we did with the fudge, is by constantly working it and cooling it quickly, those sugar crystals are going to be very small. Hard candies is another example of cooling. You cool it. Hard candies are cooled super quickly, and they're not a crystalline candy. You actually they're called like a glass, is what the even technical term is for their structure. Okay, third we have ripening. So ripening is allowing candy to sit for a period in order to form a creamy, smooth texture. And so the best example for that is fondant. So after it's made, it's wrapped and allowed to sit for 12 to 24 hours. And that allows time for the smaller sugar crystals to dissolve some, and they might sometimes also use invertase to hydrolyze those crystals. Um, and ultimately, that leads to a softer, more pliable fondant because fondant is used like this to decorate cakes, and so you want it to be soft and moldable and easy to lay over the cake without like cracking. Okay, so some tips for successful candy making, understanding the crystallization process. Um, Monitoring the concentration of your sugar solution, and how we do that is really with the temperature. The sugar concentration is going to increase as the temperature increases, and as the water evaporates, the sugar concentration and the sugar concentration increases, the boiling point increases. And so we kind of know what the concentration is just based on the temperature that it is boiling at. If the concentration is too low, the candy is going to be less hard than it's supposed to be. If so, if you're aiming for chewy, it's going to be liquid. If you're aiming for hard, it's going to be gummy or sticky. If the concentration of the sugar is too high, then the candy is going to be harder than you desired. It's going to be grainy. It's going to be dry versus what you're shooting for. Also, we want to monitor the size of the sugar crystals, kind of all the stuff that we've talked about in that last section, understanding what creates sugar crystals and how to control the size with agitation or when we are doing certain steps. Both of these are related to temperature. And so it's very important to use an accurate candy thermometer. And you'll see here on the next slide that sometimes like the temperature at which one thing happens versus the other, very narrow range, and so you need to have accuracy with your thermometer. Um, It's also important to know that high humidity can reduce the sugar concentration, especially often on the surface. Um, but essentially, high humidity can prevent candy from setting to begin with, or lead to stickiness on the surface of your candy. Um, Fudge makers generally say don't make fudge on a human humid day, because your fudge will not turn out as well. Okay, so here is a chart, and you have one kind of like this, just less color, in your book. Um, that is the names of the consistency, the temperature at which we achieve that, and what um, the candy at this temperature is used for. Okay, so the names of this, and the book does not explain this very well, the names for the stages of the sugar are based on what happens if you drop a dollop of syrup into ice cold water. Okay, so for the thread stage, it forms a liquid thread that will not ball up. So you dump it in the water, you can pull it out. There's a kind of liquidy thread, but it's not going to ball up. The soft ball stage forms a soft, flexible ball. If you remove it from the water and hold it in your hand, it will actually flatten out as it warms. Okay, so it's not really a solid. Okay, at a cold temperature, it is, but as soon as it gets warmed up in your hand, it will liquefy again. Okay, um, the firm ball stage, um, I'm trying to make sure. In the book, it just calls this ball. Okay. Um, but this firm ball stage or ball, the ball will not flatten when you take it out of the water, um, but you can flatten it really easily if you squeeze it. The hardball stage, which is not on here, but the hardball stage will form a hardball. If you squash it, you can still change its shape though, but it's not just going to be as easily flattened as the ball stage. Next is the soft crack stage. And so the soft crack will solidify when you drop it in the water into threads, and those threads are flexible, so you can bend them when you take it out, not brittle. It will bend at least slightly before breaking. If you like really bent it, then it might break, but they are somewhat flexible. For the hard crack stage, there's almost no water left in the syrup at this stage. We've heated it to 300 degrees Fahrenheit, um, or 149 degrees Celsius. And so it will form hard, brittle threads that, once you take it out of the water, you bend it, it will break. Okay, so this is the last of like the major candy stages. And then once we've gotten rid of all of the water, um, then we get into the caramel stages. Okay, so light caramel is where, like I said, all the water's been boiled away and the sugar is liquid, and a light amber in color. And then dark caramel, 20 degrees higher at 340 degrees Fahrenheit, is brown in color due to caramelization that has happened. Okay, so now we're back to our list of sugar functions. Okay, we spent a lot of time about one crystallization. Now we're going to talk about a couple other functions. First is that sugars can be caramelizing agents. So caramelization is the process of subjecting sugar to high or prolonged heat, which changes the sugar to a brown liquid. So so this caramelization is not the same thing as making candy caramel. Okay, we're not in all of these examples, or most of them at least. It's not candy caramel, but the same principle is happening, is that you have pure sugar that is somewhere um, on a crust or within the food, and it's been subjected to enough heat that that sugar turns to a brown liquid, and then when you take it out, it solidifies. Okay, um, and it might be dispersed um, like dilutely enough that like you're not gonna have rivulets of brown liquid running down your food. So, for example, the brown crust on baked goods is at least partly due to the caramelization of sugar. The beige color of sweetened condensed milk. That milk is heated to a super high temperature, and it has a lot of sugar added, and it is beige in color because of the high heat and that sugar. Okay, the color of caramel candies. Okay, caramel candy is not the same thing as this caramel. Okay, we've added things like milk and things to make it chewy. The crust on flan. So part of the way flan is made is that you have coated the outside of your dish that you're cooking the flan in a sugar solution, and then you're baking it. And what is all the sugar that is on that is touching your baking dish is getting superheated. And it actually does turn into this kind of caramel. Okay. So like I said, not to be confused with caramelized sugar stage of candy. Okay, the last function of sugar in food preparation is fermenting agents. So sugar provides a nutrient source for fermenting microbes. Um, the um, the main end products in food applications might be alcohol, it might be lactic acid, depending on what microorganisms are used. So this graph here is showing the decline of pH. These solid lines are showing the decline of pH. And the water activity, this dashed line, of sausages that are fermented with lactic acid bacteria. So the pH starts out fairly high, close to 5.6, and just the lactic acid bacteria consume glucose and they convert it to lactic acid, which lowers the pH and the water activity. And that is how we get a shelf-stable sausage, right? By just adding some sugar and some bacteria to the meat, and the bacteria convert that sugar to an acid and make it safe to eat. Okay. Fermentation of alcohol, you have sugar, yeast, you take away the oxygen, and those yeast will produce carbon dioxide and alcohol. So now we'll talk about nutrition. So switching gears and talking about nutrition and sugar. So sugar is 4 calories per gram. For when I go through each of our major macronutrients, I'm going to tell you how many calories per gram because it's a common thing that you should kind of know. Okay, so for sugar, it's 4 calories for every gram. So when sugars are digested by our bodies, they are going to be broken down to monosaccharides via hydrolysis. We've already kind of talked about that. And then the monosaccharides will be absorbed into the bloodstream. From there, they'll go to the liver, and the liver will convert fructose to glucose, galactose to glucose, glucose to glycogen. And so this glucose that is produced here will be converted into glycogen. Now, what glycogen is multi-branch chains of glucose. It looks kind of like this, and here's like a zoomed in picture. You have all the glucose here and like this. Now, why does it look like this? It's because it's so heavily branched, when we need glucose, lots of it can be broken off at once, right? We can have the same reaction occurring simultaneously in hundreds of thousands of different places. There are many more sites to break off a single glucose at the end, because think about it, each reaction is I just need one glucose to use, and you can only break off that end one at a time. And so by having all these branches, it allows you to get a lot of glucose all at once, versus if we had a straight line of glucose molecules all lined up, then it would take a lot longer. Okay. And why is it like this? We also don't just want free glucose floating around in the liver either. Okay. So the body is constantly using and replenishing these stores. And glycogen is stored not only in the liver, but also the muscles. And the glucose is just broken off and used as needed for an energy source. Okay. Now, here's a big disclaimer. I'm not a fan of this book with nutrition. And so, this big disclaimer. We do not fully understand the science of nutrition like we do many other sciences, like some basic chemistry, where it's proven so well you can count it pretty much as fact. Nutrition is super complex with all kinds of interactions between different chemicals, and no true in vitro, which in vitro means outside, inside the bottle, inside the body, like inside the living animal model, where in vitro, outside the body. So in vitro is outside the body. You can't do the science of it outside the body, because the body is, living things are so complex, and so it's very hard to understand all of the chemical processes that are going on. It is also very hard to control what people eat, and hard to measure the results of your experiments, because the reactions happen so quickly. Like I can't pinpoint exactly which reaction is causing the result that I measure, which usually the result that I measure is several steps down the line. Even if I'm measuring some metabolite in the bloodstream, well, that's not where things were digested. And if you're measuring weight loss, that's even more, hundreds, if not thousands of steps down the line. So it's difficult to connect one specific nutrient to what you see happening. Some of what we know about nutrition, and I actually put that in quotation marks, know about nutrition, it's actually based on faulty research, or just one person saying something in a research article, and everyone else just going with it and not really questioning the science. So for example, one of the nutrition studies that hugely influences how people view nutrition today is a study called the 7-Country Study. And this study was published in 1978. And this guy, Ancel Keys, he was a nutritionist. He'd heard from a friend in Italy that in Italy, heart disease wasn't a problem. And so he started studying diets in various countries and correlating them to heart disease rates. And his results demonstrated that saturated fat leads to high cholesterol, which leads to heart disease. And these are well-known facts now, right? Here's the problem. He had this hypothesis, and he was writing books about it before the study was even complete. He designed the study to some extent so that his cholesterol theory would be supported. And he purposely picked countries that would support his hypothesis. So also, one of the countries areas he chose was the Greek island of Crete. And yeah, they eat a lot of fish, but one of the 3 times that he visited there and was documenting what they were eating was during Lent. So yeah, they're eating a lot more fish than other countries because it's Lent. And yeah. So the other thing other things. Comparing 7 different countries is very confounding. If you think about it, the activities, lifestyles of people in different countries, different cultures is different. And the more variables that we insert, the harder it is to separate out the true causes of differences in what we're looking at. And so this is why actually just in general, epidemiological studies are not great. So when we take a group of people and we just survey what they ate or did and compare that to their health, there are just way too many variables for us to pinpoint this caused this. And that's kind of what ends up happening with epidemiological studies. They correlate an end result to something that they noted that the person did. And correlation does not equal causation. So let me get through some of these bullet points. So doctors also receive very little nutrition education in medical school. So a lot of the things that you're even taught by your doctor are just uneducated. And like I said, many of the well-known facts have been proven to be false. So to kind of demonstrate what I mean about correlation does not equal causation, I'm going to present these graphs. They're kind of fun. Okay, so this is the number of people who drown by falling in a pool each year from, I can't tell what that says, 1999 maybe to 2009, what it looks like. In red, and the number of films Nicolas Cage appeared in black. They're correlated. Nicolas Cage is killing people in pools. Yeah. Another one that's a little bit more serious, but showing you how not right. Supposing that things cause something just because they're correlated are so years 1998 to 2007, the organic food sales were spiking at the same rate, almost identically, to the autism diagnoses in that time period. Man, do we really think organic food sales is causing autism? No. So you have to be very careful when studies tell you something is correlated with something else. So in kind of overall conclusion of this slide, the field of nutrition is ever evolving, and there are many nutritionists in the field that even now, like that Ansel Keys study is still used today to tout the fact. And there have been studies that, despite there being studies that do disprove his cholesterol theory to some extent, it's still like what people know and what people go to. There are many nutritionists in the field that just go with the status quo and lean on research that might be faulty, but there are some great up and coming nutritionists that are really challenging that status quo. But the overall comment there is like the field of nutrition it's murky. And I disagree with a lot that's in the book. I could be wrong too. But I think that science is questioning things, and yeah. Okay, so from the book, talk about what we need glucose for. And I have little asterisks here because these are things that I disagree with the book on. So full disclosure here, I am on a semi-ketogenic diet. I don't do it super strictly. I still indulge, but I do limit my carbohydrate intake for the most part. And I personally believe that carbohydrate needs are much less than the book states or most people believe. I encourage you to do your own research into this. But I'm going to try to give it from both perspectives and share also what I know. Okay. So metabolism of fat and protein. So the main metabolic pathway for fats and proteins does use glucose, but Ketones, which are produced in people consuming low carbohydrate diets, can also be used for this metabolism. Um, and so some of the common knowledge nutrition says that without carbohydrate intake, our bodies will produce glucose by cannibalizing our lean muscle tissue. And it's going to, because we need to, well, it'll have to use that protein to make glucose. And this process is called gluconeogenesis, which means glucose, new creation of new glucose. But actual studies that have tested this have shown an increase in lean muscle mass on people on those diets. And it'd be true if you don't consume enough protein, you're going to be in trouble. But as long as you're consuming enough protein, you're fine. And the body adapts. And honestly, who's to say, in my opinion, like, who's to say that the high carbohydrate state is what is actually normal? Maybe the ketone state is. I don't know. Okay. Also need glucose for brain function, but actually the brain thrives very well on ketones. There's actually studies that show that if the brain has a metabolic disorder, which can be found in diseases including epilepsy, mental illness, even mild cognitive impairment, a low carbohydrate diet such as a keto diet can help. And what happens in the brain is instead of using glucose, the brain uses ketones, and it does very well on that. That's kind of how the ketogenic diet was even discovered. Was somebody was studying epilepsy and found that it helped the brain. So, but a lot of nutritionists will tell you that it's very bad for your brain, but it's not really true. Okay. And I get a little bit passionate about this nutrition stuff, but okay, so some negative effects of sugars. This isn't listed as a negative effect in the book, but serotonin, increased release of serotonin. So serotonin isn't a bad thing. It's a chemical with a calming effect and makes you sleepy. But I don't know that it's like a great thing to feel sleepy after every time you eat a bunch of sugar. I don't know. So sugar coma is a real thing. And it's because sugars cause the increased release of serotonin. Now, big discrepancy with the book. This book tries to tell you that one sugar is no different than another, and they're wrong. So some sugars are worse than others. Okay, this is a fact. And the liver does metabolize fructose and glucose differently. So yeah. There's lots of red here. Okay. So fructose along with a high fat diet, there have been studies that have shown that that does lead to metabolic dysfunction, increased total cholesterol, and increased blood triglycerides. So triglycerides is like fat in your blood, right? And it's a measure of heart health. Metabolic dysfunction is, we'll talk about insulin in a minute, but reduced tolerance to glucose, impaired insulin signaling in your body, and having lowered insulin sensitivity for that. Okay, but I would say that perhaps more important than how much fructose you consume is how fast fructose is consumed. So going back to last week, the high fructose corn syrup 55, so it's 55% fructose, is what's used in soft drinks. Now this isn't a huge difference in the amount of fructose versus fruit. For example, apples, depending on the variety, range from about 52 to 69% fructose. So it might even have more fructose than the amount in a soft, well, ratio wise in a soft drink. But like amount wise, like one Coke has as much fructose as 3 oranges. Okay. How fast do you eat 3 oranges? Right. Like, and I'm not talking about the little ones, right? Like 3 good-sized oranges. How fast do you eat that? Versus how fast can you chug a soda? Um, and and not only that, but fruits have other nutrients and they have fiber and they have things that slow down that digestion. And so, how fast is that soda sugar going into your bloodstream versus eating fruit? Um, and um, and so you're getting this like huge shot of all sugar, but including fructose, really fast. Um, the the liver does process fructose differently. It has to turn it into glucose first, right? And there are certain enzymes that do that. Um, Also, um, some of it we don't know why fructose causes some of the metabolic issues. But we do also know that fructose does not turn off hunger signals like glucose does. And so there's a tendency for more overeating with the consumption of fructose. And so with the soda, what I would say is bad about this versus an apple is you are getting a huge shot of fructose all at once, and your liver has to work harder to process it than it does glucose. And it's, I think, probably part of the issues that come from high fructose is that the liver doesn't have time to process all of that coming in on it at once. Um, Versus fruit, it does. It comes in more slowly. It's tempered by number one amount, but also how fast it's absorbed by the body. So now that being said, fruit juice, you've eliminated all the good things about fruit by putting it in a juice because you have eliminated all the fiber there and you've processed it. And so the sugar from fruit juice can be considered really just as bad as soda because it's going to hit your bloodstream just as fast. So lest you think you're doing a good job drinking OJ in the morning and not drinking soda, I hate to break it to you, but yeah, orange juice is pretty much as bad as a Coke. Okay. Uh, so other negative effects of sugars, tooth decay, dental caries. So I've already talked before about how acid damages enamel. That acid can be sourced from food or from bacteria. And how bacteria produces acid is first, they like sugar. And it can be any sugar. It can be sucrose, it can be fructose, glucose, but it also can be starchy things. So bacteria like crackers just as much as candy. Um, so they like the sugar. Then as they grow, they produce what is called a biofilm, which we know on our teeth as plaque. And it's this hard wall that the bacteria are using to protect themselves and grow in and be happy, that's preventing anything from easily brushing it off. Okay. Um, that biofilm creates an anaerobic environment without oxygen. And when anaerobic, certain bacteria that like no oxygen are growing, they will use that sugar and produce acids, kind of similar to the fermentation of sugars that we talked about several slides ago. Okay, now these acids are on your teeth and um, decaying your teeth. So overall, here I say like eat, rinse after eating sugary or starchy foods, brush if you can. Um, eat sugary foods infrequently. My dentist told me once that like nursing a soda all day and just like sipping on it all day is far worse than chugging it for your teeth anyway, right? For your health, chugging it is worse because you're getting that sugar shock. But for your teeth, so just don't drink soda. Limit the acid beverages, period. So OJ, right? Sugar and acid, really bad combo. Chew sugarless gum after meals. So cool thing about sugarless gum is most of it contains xylitol, and xylitol is actually really good for your teeth. And there's scientific studies that have shown that it reduces existing plaque. So, chewing gum is actually a really good thing to do, especially right after meals. Okay. Final think negative effects of sugars. There's one more after this. So diabetes, mellitus. So it causes the body's inability to move glucose from the bloodstream to the cells. So we have insulin receptors on our cells, and insulin is produced by the pancreas, and it triggers glucose to be taken up by the tissues. So our muscle, for example. So muscle cells have insulin receptors. Insulin binds to it, and the muscle says, "Okay, I'll take some glucose now," and allows the glucose to come in. And so the glucose is then used by the tissues for storage of an energy source. But diabetics do not produce enough insulin. Type 1 or type 2, they don't produce enough insulin, sometimes type 2, type 1 always. Or the cells have insulin resistance and don't respond to the insulin signals. So the insulin is like knocking on the door of the muscle, hey, I've got some glucose for you. And those receptors are asleep because they've been overstimulated, and so they're ignoring it, and glucose. And in the meantime, so this is that's type 2. In the meantime, the glucose is just hanging around in the blood and causing damage. So high blood glucose can lead to nerve damage and poor circulation. And so these two things are why amputations are a serious and common threat in diabetics. So especially the lower extremities, blood doesn't travel well to those extremities, and ulcers are more likely. And then if the ulcer is very severe or it gets infected, then you often have diabetics losing toes or feet or things like that. So of about 200,000 people who have amputations each year, 130,000 of them are diabetics. And if we take out the traumatic injury related amputations that happen each year, diabetics account for 80% of lower limb amputations in the US. So diabetes is a scary thing. Now, type 1 diabetes is an autoimmune disorder. It has nothing to do with the person's diet as far as the causation of it. Like their diet's very important once they have it, but and controlling their diet so they don't have issues of high or low blood sugar for them. But type 2 diabetics is generally going to be caused by the person's prior diet, their sedentary lifestyle, obesity, etc. So especially important for type 1 diabetics, low blood glucose will lead to dizziness, shaking, confusion, and can cause some serious issues. It's just as dangerous for them to have low as high blood glucose. Blah. So, okay. In general, diabetics diet-wise should avoid sugar spikes. So they should not eat super high sugar things. They can have carbohydrates, but it needs to be more of like whole foods where the carbohydrates are digested more slowly, so they aren't getting big sugar spikes like from drinking a soda. Um, so. Okay, last one, weight gain. I have more to be upset about here. Okay, so I don't like the part of the book about weight gain at all. In fact, I'm gonna say just don't even read it. Okay. Ugh. Okay. Weight gain. So the book doesn't say this exactly, but they're kind of saying it, and not this phrase, but there's a common phrase that doctors will say is a calorie is a calorie is a calorie. And what they're basically saying is like calories in, calories out, and you should only be paying attention to the calories that you eat. And the exact sugars don't matter and all that. Not true. Okay. This would actually violate the second law of thermodynamics, that entropy or disorder must increase in all reactions. And big C calories are determined by combusting the nutrients, like literally putting them in a fire and burning them and measuring the energy that is produced. And we are not combustion ovens. Different foods go through different biochemical pathways, and some of those pathways are more efficient and some of them are less efficient. And so some of those calories are lost as heat and not actually going and stored as energy in our body. So not all calories are the same. So energy lost in digestion. So it takes energy to digest foods, and it takes a lot less energy to digest fat or carbohydrates than it does protein. We lose about 25 to 30% of the calories of protein, due to just the metabolic processes it takes to digest them. Okay. Um, so in addition to this, sorry, let me go. So different nutrients cost our bodies more energy to digest. This high protein diet is going to take much more metabolic energy to take in versus a high carbohydrate diet. Hence, we'll say eating more protein boosts your metabolism, because calorie for calorie, if you're just looking at calories, you are going to use up more energy digesting protein. Whole foods also take more energy versus highly processed foods. Also important, different foods and macronutrients have a major effect on the hormones and brain centers that control hunger and eating behavior. I've already described a little bit like how fructose does this, for example. Different foods are more satiating. So things like donuts, cakes, candy, they're not satiating at all. They don't make you feel full. But beef, eggs, beans, even fruits are more satiating than those things. On my keto diet, when I'm doing good following it, I am not even hungry in the morning, and I've always been a breakfast person. But like I honestly have to make myself eat to get enough nutrients sometimes, because like I'm very easily not hungry anymore after I eat. Also, if we're talking about sugars, so eating high levels of sugar all at once can lead to rapid spikes in blood sugar, and then lead to a crash in blood sugar later, right? And this causes cravings for more sugar. And so eating high levels of carbohydrates that are easily and rapidly digested, such as refined grains, and sugary foods, if you're not eating those in moderation, you're more likely to become obese and diabetic, because of those blood sugar spikes. What kind of happens is those insulin receptors become resistant to the insulin, and you get that before you get full-blown diabetes. happy to talk about this more in depth with anybody. I'm not a nutritionist, I'm not an expert, but I know enough to feel like I'm educated about it myself. So, sorry this went a little bit longer than normal this week, but yeah, there's a lot to cover and some things that I'm a little bit passionate about. All right, y'all have a great week.